The overall goal of this procedure is to purify virus-like particles, or VLPs, expressed and secreted by mammalian or insect cell expression systems.
This method can answer key questions in the vaccinology field, such as how to express and purify conformationally relevant viral antigens in a safe and flexible manner. The main advantage of this technique is that it does not require protein precipitation using polyethylene glycol or preparation of multiple density layers to concentrate VLPs.
Though this method can provide insight into the identification of viral antigens as vaccine targets, VLPs can also be used as tools for disease diagnosis and serology.
Visual demonstration of this method is critical as the glycerol underlay and VLP resuspension steps are difficult to learn because beginners may fail to exercise proper technique.
On the day of transfection, prepare liposome and DNA solutions per the manufacturer's recommendations. Transfect the DNA cells with a DNA composition of 1:1:2 of HA to NA to GAG and a total DNA quantity of 40 micrograms per T150 flask. Repeat this step to create 9 T150 flasks for a total volume of 200 milliliters. Next, dilute the DNA and liposome solutions in serum-free transfection media without antibiotics so that each flask contains a total volume of 24 milliliters. Return the flasks to the incubator and maintain the cells in transfection culture medium until the day of virus-like particle, or VLP, harvest. Transfer the supernatant into 50 milliliter conical tubes to harvest the culture from the cells after 72 to 96 hours post-transfection.